Compared to other European countries, Ireland has doubled the amount of newborns with spina bifida. Around 2/3 of overall cases of neural tube defects can be prevented through periconceptual folic acid fortification. Therefore, national plans for folate fortification and supplementation should be urgently addressed. Folate is one of the B vitamins. It is a water-soluble vitamin that can be found in many foods, such as green leafy vegetables, citrus fruits, beans, and cereals. Folate cannot enter the metabolic pathways if not previously reduced to tetrahydrofolate. This reaction is carried out by DHFR, an oxidoreductase that uses NADPH as a cofactor. Tetrahydrofolate, which is the active form of folate, is the most versatile one-carbon donor in biosynthetic reactions. After identifying the fundamental role of tetrahydrofolate, DHFR was extensively studied and targeted with chemotherapy drugs. DHFR was thought to be the only functional one of its gene family until 2011, when a second DHFR-like gene was discovered and called DHFR2. This previously reported pseudogene is now known to be an expressed retrogene in humans and has opened up a new set of questions about its involvement in folate 1-carbon metabolism and its interplay with DHFR. Our project is about interrogating the function of DHFR2, which has proven to be a challenge due to its strong similarity with DHFR and its low translation rate. Imagine DHFR and DHFR2 are like a peach and an apricot. Can you easily identify DHFR2? To solve this problem, we are generating a DHFR homozygous recessive cell line using CRISPR-Cas, aiming to unequivocally identify DHFR2 and its function. Within this experiment, we also aim to assess if DHFR2 is able to compensate for the lack of DHFR. DHFR2's protein has not been detected so far. The reasons for this could be its low translation rate or its similarity to DHFR, making traditional methods difficult to use, it not being translated, or it being tissue specific. Due to these reasons, we are using a state-of-the-art mass spec in line with the targeted approach to determine the presence or absence in numerous cell and tissue types. Finally, we are investigating the increasingly wide pool of DHFR2 RNAs through bioinformatic analysis and PCR confirmation. From what we have discovered so far, different sets of isoforms are expressed depending on cell type and proliferation differentiation status. To conclude, discovering how DHFR2 works will translate into a broad range of possible applications, from the design of new chemotherapy drugs to alternative supplements for the prevention of neural tube defects.